Welcome! I'm Andy, and I'm here with Tim.
Hello.
So this is your study guide for Exam 2 for Paleobiology spring semester. So this exam will cover just the material since Exam 1. So we'll go through our 8 topics, which is the Ediacaran fauna through arthropods. So this study guide will cover the Ediacaran fauna, Cambrian explosion, sponges, cnidarians, brachiopods, bryozoans, mollusks, and arthropods. So I hope that this video will give you a good idea of what to review for the exam. And then please come in to class on Wednesday with any questions. So that class on Wednesday will be entirely devoted to your studying. And then the exam will be on Friday. So let's get going. All right, so the first topic that we covered after the first exam was the Ediacaran fauna. So the Ediacaran fauna was a group of animals that lived in the very end of the Precambrian, so from about 575 to 542 million years ago. It was a mostly soft-bodied fauna preserved as impressions in sandstone, and these beds were often storm events. And they're often associated with algal mats. So most of the fauna was soft-bodied, but there were some mineralized skeletons. We talked about Clodinia, which is this simple calcite tube that had drill holes in it. So that was evidence of predation in the Ediacaran. But most of the fauna were these large, flat animals with lobes that are soft-bodied and only preserved as these impressions. There was also sponge spicules preserved, so their sponges had evolved by the Ediacaran.
But no body fossils for sponges?
Not that I'm aware of. So we get our first body fossils of sponges in the Cambrian explosion. But that's a good point, Tim. We should check on that. We definitely have sponge spicules. I'm not sure about body fossils. So these Ediacaran fauna were both radially and bilaterally symmetrical. So we went through a list in lecture of some of those fossils. So you don't need to memorize the lists of those fossils. It's interesting, but that's kind of this stamp collecting. But we don't have to go through all the list of the fossils. But just know that some were radially and bilaterally symmetrical. There were also those big frond animals, Dixonia, that we saw at the Cleveland Museum of Natural History. Those had no mouth. So we think they had endosymbiotic algae that was producing nutrients for them. So the phylogenetic relationship of the Ediacaran fauna to today's animals or metazoans is unclear. So it does seem like there were some basal cnidarians, so jellyfish-like animals around in the Ediacaran that may have survived, and there may even have been some basal arthropods, so arthropod-like animals in the Ediacaran. But most of the Ediacaran fauna doesn't resemble the modern fauna at all, so we're not exactly sure what happened to these things.
It's a hot topic. You should find, with a minimum amount of effort, some very contemporary work in popular science and also in the major mainstream science journals surrounding this topic. It's really interesting and very, very poignant as of right now.
Yeah, so we read that article that was arguing that the Ediacaran fauna may have been actually on land, but that's highly uncertain, and we discussed some of the problems with that hypothesis. We don't really know what these things were. They might have been ancestors of cnidarians or arthropods. They may have been they may have been giant protists or maybe lichens, which is a fungi-algal mutualistic relationship. Or they may have been, many of them may have been actually a sibling group to metazoans. So this is a branch of life that has left no descendants. So we're not exactly sure what happened to them. But they were driven, there was a mass extinction of many of these things at the beginning of the Cambrian. So they may have been outcompeted by the Cambrian organisms that we'll talk about in just a minute. So, but going back to the Ediacaran fauna, they were distributed worldwide, and we talked a little bit in lecture about how they may have been distributed so across the world because there were these very productive algal mats, and there was probably very little competition from anything else. There seems to be no geographic provinciality to them, so it's not like we see different faunas in different parts of the world, but rather we do see different communities distributed according to sedimentary environments. So we see some associated with these deltaic environments and others in these shallow shelf environments.
So cosmopolitan species assemblages.
Right. So what does that mean, Tim, cosmopolitan species?
It means they are, like you said, broadly distributed with no defined geographical boundary, and in the case of the Ediacaran fauna, it's likely that there was no real geographic element, so that it was mostly environmental. But just another way of saying what you said.
Yeah, cool. Thanks, Tim. So what was the ecosystems like at these in the Ediacaran? Well, there was these abundant algal mats that left the stromatolite records, and there was a fixed and mobile benthos, so there was sessile organisms at the benthic organisms, and there were also some mobile benthic organisms that crawled along the bottom. And there were some nectonic swimmers, so these swimmers may have been the ancestors of modern-day jellyfish. There were also filter feeders and algal mat grazers. So that's all I have to say about the Ediacaran fauna. It's just enigmatic organisms that are hard to understand. So.
Different interpretation of there are different interpretations of the Ediacaran fauna depending on primarily what you consider the depositional environment to be. So you've talked about the controversial terrestrial hypothesis, which we say with a little chuckle. The shallow water hypothesis, but I've read a lot about deep water. The problem with the deep water hypotheses is that the frond-like organisms couldn't have been photosynthesizing. So there are some chemosynthetic hypotheses about how they survived. But again, it's a very interesting subject. A lot of it's speculation. A lot of people trying to extract as much information from a very sparse record of that particular point in time. But it's interesting stuff.
Yeah, it's very enigmatic. We're not quite a puzzle. Yeah. But so many of them were probably driven to extinction by the Cambrian organisms, which appear at about 542 million years ago. Thanks. So that is the start of the Cambrian, and it's also the start of the Phanerozoic, which is the age of visible life. So the small shelly fauna that we talked about, which was the advent of skeletalization. So these were small shells that are difficult to classify to modern groups. Some of them may have been mollusks. Others may have been parts of larger organisms or parts of mineralized parts of sponges or mineralized parts of other organisms. So this small shelly fauna marks the beginning of the Cambrian. So these are important biostratigraphic markers for the beginning of the Cambrian.
But enigmatic again.
Yeah. Yeah. Enigmatic. We're not sure exactly what kind of organisms produce this small shelly fauna. But the Cambrian explosion is well documented in the Burgess Shale and other even younger or even older deposits. such as the Cirrus Passat in Greenland and then Xinjiang in China. But we talked a lot about the significance of the Burgess Shale fauna. So that was at about 505 million years ago. It's preserved in the Canadian Rockies. It has exceptional preservation, so we get soft part preservation due to this rapid burial and probably with anoxic conditions that prevented decay.
It's hypothesized that these animals were shelf dwellers, right? And localized, but rather large mudslides were happening, and sweeping down the side of the shelf, moving all of these organisms and burying them rapidly. So we had a collection. You have some in situ and some ex situ faunal elements. So that's the hypothesized deposition environment anyway, right?
Yeah, that, just as you said, there were these shelf-dwelling environments, and there was this big sediment slump that deposited...
Slump, that's the word I was looking for.
Slump!
Damn it.
That deposited them in deeper water that was anoxic, and then they were buried very rapidly. So these are organic shales, and a lot of these organisms are preserved as imprints in this organic-rich shales.
Carbon films, but there are three-dimensional. There is three-dimensional preservation, but because there is so much compaction, it took a while for researchers to figure out the three-dimensional preservation.
Yeah,
but
definitely.
It's been very valuable since it was figured out.
Oh yeah, the first researcher, Walcott, he didn't grasp that these were three-dimensional fossils. But then there was some really evolutionary breakthroughs when...
Was it Conway Morris that did it?
And Winningham and Briggs,
yeah.
Who actually were able to very finely dissect these carbon films to see the morphology of the organisms in 3D. And that's when we
got a better idea of what it is that we were looking at.
Yeah. Okay, so what are the Cambrian organisms? So we see body fossils of sponges. We see lots and lots of arthropods. We see segmented predators, those strange flower-like animals that had that were attached to the sessile benthos, and they had a mouth and an anus with the petals all around it. Yep. We see maybe velvet worms. What are those?
In the Cambrian fauna, so the modern representatives are onychophorans.
Yeah, that's the word.
But in the Cambrian fauna, they're most likely represented by the basal group, the lobopods.
Okay.
They're both part of the same group, but onychophorans are more derived. The lobopods went on to form some pretty funky morphologies. It's worth looking them up.
So lobopods, we talked about hallucinogenia. That may have been an early lobopod. There are also some early mollusks and an early chordate, Pacaya, which is perhaps is definitely the first representative in the fossil record of our own phylum, human chordates.
Half fish, half invertebrate, looks like.
Yeah.
It's got the muscle blocks of a vertebrate at the back, but it's got a weird kind of snail head.
Yeah.
It's weird.
It is, and it has the long notochord that is characteristics of the chordates. So a lot of weird animals, and many that are very difficult to classify. But the Cambrian explosion is significant because we see the first advent of large body fossils of organisms in the fossil record. We see the evolution of skeletalization, which allows for increased defenses from predators and also as the site of attachment for muscles, and also allows the sequestration of certain elements that you don't want in your body. You can sequester them in these skeletons.
And very considerately of the organisms, it allows them to be preserved more readily.
Very considerate, yes.
Yeah.
So we can study them.
Yeah.
And so we can...
Very thoughtful.
Yeah, so we can talk to you about them. So we see the range of new body plans, and we see the evolution of all major phyla of animals except the bryozoans. So animal phyla correspond to basic body plans. So, and in the Cambrian explosion, we see the evolution of all major body plans of animals that are alive today, except bryozoans, which evolved in the Ordovician.
But
Yeah.
but brachiopods were around in the Cambrian,
Yes,
and they are both lophophoridates.
they are both lophophoridates.
So does that mean that bryozoans were absent? Were they just absent from the assemblages we have, or did they definitely not originate then?
Well, so the Cambrian has been extensively sampled for fossils. So if there were bryozoans in the Cambrian, I think we probably would have found them.
To see them, yeah.
So that suggests that this origination in the Ordovician is really a true, they just evolved then, and they weren't around in the Cambrian. I don't know, what do you think?
I tend to agree, but I wouldn't be surprised if they existed in a soft-bodied form and developed calcification at a later date. That would be just as plausible for me. But you'd expect in Burgess shale-type fauna with soft part preservation to have some trace.
Right, that's what
Unless they started as individuals and developed coloniality later on.
We're getting a little ahead of
Sorry.
ourselves, but bryozoans are the only phyla of animal that are exclusively colonial. So what Tim is suggesting is that maybe they originated as individuals and then only later evolved coloniality.
Well, they had to have originated as individuals, right?
Yeah.
Anyway,
That's interesting.
Kind of thoughts that you should not be afraid of having.
Bryozoans are unique for a lot of reasons. So we see the evolution of skeletalization, the extinction of the Ediacaran fauna, and more complex food webs developing. So in the Ediacaran, we had at least predators, herbivores, and the base of the food chain, which are autotrophs. So we had algomats that were autotrophic, herbivores, these algal grazers that ate the algae, and then we had at least some predators because we see the evidence in the boreholes in Claudinia. But in the Cambrian, we see more diversity of predators and more complex food webs. We also, in addition to the evolution of the major body plans, we see the evolution of other body plans that have left no descendants. So we read this Stephen Jay Gould article that argued that the disparity of life was very great during the Cambrian. Here's my hands. There they are. So we see lots of body plans in a larger difference between body plans alive in the Cambrian than we see today. So we see more disparity in the Cambrian than we see today. And then only some of those organisms survive to leave descendants. So many of these body plans that aren't around today were kind of failed experiments that left no descendants. And so Gould argues that it was kind of a lottery process, which body plans survived to leave descendants. And the phyla that we have around today is a legacy of those Cambrian organisms that left descendants.
There is when we talk about the lottery element of that analogy, we are talking about environmental changes that occurred that favoured certain taxa over others.
Well, Gould would argue that it's not necessarily
There was no selective process.
Right, that it was random. That only a certain number of those body plans could survive. So this is a debate that's still ongoing. So other people dispute the idea that there was a larger disparity of body plants in the Cambrian and say that those organisms, while they may look strange to us, were in reality no more different to each other than animals that are alive today.
Yeah.
And so what caused the extinction of those those organisms with different body plans is not clear. So was it really random, or was, as like Tim said, there were some selective pressures favoring those body plans that left descendants? So that's a good question. Okay, so what caused the Cambrian explosion? Various what caused the rather sudden appearance in the paleontological record of all these taxa with skeletalized remains? So
It wasn't magic.
no, good point. So there were environmental changes at the time. There was a rise in atmospheric oxygen, but that happened earlier, so that's not a good candidate for a cause. There was higher temperatures and increased area of continental shelves because of tectonic processes. So that leaves more very productive continental shelf environments for life to evolve. So that could have played a role. At this time, there was also an increase in calcium in the world's ocean, the cause of which is not properly well understood. So that probably also favored the development of these skeletalized bodies. Other suggestions for causes include an arms race. So if you have increased predators, the prey is going to evolve more adaptations to avoid predation, and that causes the predators to evolve better adaptations to capture prey. So there could be this predator-prey arms race favoring skeletalization. It's also argued that this is just a taphonomic artifact, that animal phyla actually evolved earlier in the Ediacaran, but they were small and inconspicuous, and it wasn't until the Cambrian where we get this biotic arms race, which causes an increase in body size and skeletalization, which is why we see the fossils in the record at the Cambrian.
There's also the possibility that the depositional environments required for fossilization may not have been prevalent in the Ediacaran. If you think about how many different biogenic components need to go into a preservationally favourable environment, such as excess calcium, which hadn't risen until then. So the chance of being preserved before the Cambrian may have actually just been lower due to environmental factors.
Right, exactly. So that's another good point.
I'd like to also add that the increased area of shelf this is likely in my mind because it's also been proposed for other periods of increasing diversity in the Mesozoic. With the breakup of when continents break up, you increase the surface area of shelf that's available to colonise, and that coincides with increases in biodiversity globally.
And it coincides with an increase in the origination rate for many taxa. So that's a really good point. We also talked about Hox genes, which are genes that control the basic body plan of animals. So we see increased number of Hox genes in more animals with more complex bodies. So it could be that early in the Cambrian, the Hox genes evolved, and different lineages got different numbers of Hox genes, which increased the range of body size and made animals with more Hox genes more complex.
It kind of increased plasticity. The idea of having surplus body segments to play around with, if you will, increases the adaptability to environment. In my mind, I think it's one of the most key elements of metazoan evolution. Probably plays a large role, if not defining role, in this phenomenon.
Yeah. So we talked about arthropods the last few days, and they, because of their plasticity and how they can arrange their body segments due to their Hox genes, that's one reason why they're so evolutionarily successful.
It's like if we had an extra pair of arms, we would reduce the selective pressure and therefore the constraint on our hands being this particular design. If we have one pair of hands that are this design, we've got that base covered, and it would give us more room to adapt the other appendages to do a different selectively advantageous application.
Right, and we see that with arthropods when their appendages are modified for different functions like locomotion or digestion.
Sensory appendages like the antennae of arthropods, we'll come to this later, have evolved from different pairs of appendages multiple times.
Yeah. Great. So we will talk about arthropods later. So that's all I've got to say about the Cambrian explosion. So let's move on to talking about the first phyla of animals that we discussed in lecture and in lab, the sponges, or porifera. So molecular evidence supports the idea that sponges, porifera, is a polyphyletic. It's not monophyletic, so we see different lineages in sponges, but we lump them together because of their morphologic similarity. So porifera are the first metazoans. So what is a metazoan? A multicellular animal. So metazoans mean multicellular animals. So they're defined by being multicellular eukaryotes, capable of locomotion at some stage of their life. So even though sponges are sessile, they don't move, they're attached to the benthos, their larval stage is planktonic, so they do locomote at some stage. Metazoans are also heterotrophic, and they have specialized cells, so their cells specialize in different body layers. So sponges are the simplest metazoans. They are loosely integrated cells with porous skeletons. They're filter feeders mostly. There are some carnivorous sponges that capture fish and small arthropods, but for the most part, sponges are sessile and filter feed. But they lack true organ systems. So their basic biological organization is at the cellular level. So they are exclusively aquatic, found in many different environments, including coral reefs, mangroves, seagrass, and other environments. They exhibit either radially symmetrical or not symmetrical at all. They reproduce by budding asexually, and they also reproduce sexually to form these larvae that are planktonic and then settle out into the benthos to grow into an adult sponge. They can filter up to 1,000 liters of water every day. So they bring in water into their pores, which are called ostia, and using their choanocytes. So choanocytes are a type of sponge cells that bring in water by beating their flagella, bring in water through the ostia, and then the sponge extracts nutrients, so extracts microorganisms and nutrients from the water, and then pumps out the water through their hole in the top part of the sponge. So there's all these...
Is the hole the operculum?
Well, I forget what the hole at the top of the sponges is called, and that means we will not ask you on the exam.
I can look it up in the book.
Yes. We should look it up. So good point. Thanks. Hole in the top. So most sponges are organized as a hollow cavity with pores called ostia, and then a opening at the top. So they pump water in, and then they shoot the excretory processes out the top, and also they shoot their sperm and eggs out the top as well.
And they mass spawn, right?
They do.
Broadcast spawn, so both male and female gametes are released into the water column at the same time, so they can find each other and have little baby sponges.
So great. Tim is now going to look up what the hole in the top of the sponge is called. Thank you, Tim.
No problem. My pleasure.
All right. So choanocytes beat water in the ostia. There's also amoeboid-like cells that are undifferentiated and have various reproductive or excretory or skeletal functions. Epithelial cells provide skeletal support along with mineralized spicules. So the spicules are mineralized parts of the sponges that hold the sponge together, and they're really important for taxonomy of sponges.
The opening is an osculum.
Osculum, the opening at the top.
Small ones are ostium. The big one at the top is osculum.
So you got ostia, small, all around the sides, and then the osculum at the top.
And the cavity in there is the spongocele.
Okay,
Ta-da!
great. Thank you, Tim.
No problem.
So we talked about 3 classes of modern-day sponges. And so these 3 classes are distinguished based on the mineralogy and the morphology of their sponge spicules. So the glass sponges, or hexactinellidae.
Hexactinellidae, yeah.
Hexactinellidae, thank you.
Nailed it.
Glass sponges have spicules made of silica. Calcarea, so the class Calcarea have calcite sponges, and the demo sponges, which is the most diverse class of modern-day sponges, have organic spicules made out of the protein spongin. So we've got three different types of spicules: silica, calcite, and spongin. So silica and calcite spicules will get preserved in the rock record, and then spongin spicules are unlikely to be preserved because they're made out of protein, so they'll be quickly degraded, but sometimes we'll see them as impressions. And we did see some impressions of spongin spicules in lab. We also talked about the extinct archaeocyathids, which are sponge-like organisms. They're kind of intermediate in their organization between sponges and corals. They were important Cambrian reef formers. They were important reef formers in the Cambrian. They're entirely extinct, though. They had those double walls. So sponges through time have been important reef formers, along with the corals, which we're going to talk about now. We're going to talk about cnidarians, which is the phyla that corals are in, but this phyla of cnidarians also includes jellyfish, hydras, sea anemones. So porifera means pore bearers in Latin, so they're named for these ostia, and cnidarians mean needle bearers. So they're named for their stinging cells, which are common to all cnidarians, and they are predators using these stinging cells to capture prey.
The stinging cells are nematocysts.
Nematocysts, needle bearers. So cnidarians are basal eumetazoans, so they're distinguished from the sponges or porifera because they have true tissue layers. So their cells are organized into tissue layers. So they have layers of their body that are specialized into various functions, such as nervous systems, muscular tissues, and reproductive systems.
They have no gut, no brain, and no respiratory system.
They have no gut. They have no true gut and no brain. They have no discrete organs, and they have a simple two-way digestive system. So they have the one opening that serves to take in food and then excrete waste. So that's pretty
No, thank you.
gross if you think about it. They have a two-way digestive system. They exhibit radial symmetry, and all members of the cnidarians spend some of their lives in this mobile medusa stage and this sessile polyp stage. So some cnidarians, like corals, spend most of their lives at the sessile polyp stage, and then others, like jellyfish, spend most of their life as this mobile medusa phase. They reproduce both asexually and sexually, and many members of this phyla are colonial, giving us these spectacular coral reefs.
And also the deep-sea siphonophores, with the curly colonies that free float in the ocean.
Cool.
I love them.
So, let's see. Medusa release the sperm and egg, and those join to form larvae. So that's how they reproduce sexually. So jellyfish lack hard parts, but some are preserved, and we do see some jellyfish-like organisms in the Ediacaran. But in terms of paleontologically important cnidarians, the most important are the class Anthozoa. So Anthozoa contains corals, both calcareous corals that leave a really great fossil record, and some organic corals that don't leave such a great fossil record. So Anthozoa are exclusively marine, and we talked about some kinds of corals. So in the class Anthozoa, The subclass of Zoantharia contains the order Tabulata, rugosa, and Scleractinia. So tabulata are the tabulate corals. These are extinct colonial corals, and they, remember, had the tabulae running between the septa. So we saw
horizontally. So the septa run
vertically.
Vertically, thank you. And then the tabulae will run horizontally.
Yeah. So tabula is Latin for little tables. So these tabulate corals have the tabulae. And they were major reef formers in the Silurian through the Devonian. Rugose corals are horn corals, the common name. These are some solitary and others colonial, and these were important reef formers in the Paleozoic, but they went extinct at the end Permian mass extinction. So the only corals that we have around today are in this order Scleractinia. These are the hexa corals, and they came to dominant reefs in the Mesozoic and the Cenozoic. So these are our modern corals that you'd see if you went out snorkeling in the Caribbean or some other wonderful tropical environment today.
The individuals of the colony actually have the 6 hexagonal symmetry.
Yeah.
Not the colony overall, but the individual polyps.
Right. So the individual polyps add their septa in multiples of 6, unlike the rugose corals, which add their septa in multiples of 4. Modern corals are only found from about 30 degrees north to about 30 degrees south. So they need this warm tropical water to survive. And they also need not a lot of triginous material, so the water needs to be pretty clear. Because many of these corals have symbiotic algae that live inside them, and they need the
symbiotic
light too.
It's quite the mouthful. Symbiotic photosynthetic zooxanthellae. So they're the specific parts of the individual polyp that contain this photosynthetic algae. They have a mutualistic relationship.
Yeah.
Everybody has fun.
Mutualistic means both organisms benefit, so both species benefit. The coral benefits because it gets nutrients from the algae, and the algae benefits because it gets protection. The coral provides protection, so a sponge doesn't filter feed and eat the algae, for instance, or some other animal doesn't come around and eat the algae. Okay, do you have anything else to say about cnidarians?
Apart from they are awesome, and I wish we get more preserved jellyfish. No.
All right, so they're an important part of the fossil record, especially the corals. So we're now going to move on and talk about lophophorates. So lophophorates have a lophophore. The 2 phyla that we talked about are brachiopods and bryozoa. So lophophore is a specialized feeding organ forming a ring or crown of cilia. This may be modified into other shapes later, but it extends out of the animal and then rocks back and forth to filter feed. So we talked about the whip your lophophore back and forth. So it extends out of the animal and then it filter feeds using this specialized feeding organ called a lophophore. So the lophophore, these are the first animals that we're talking about that are bilaterally symmetrical. So they can have one plane of symmetry.
They can stand in a mirror like that to make a hole.
So the sponges were either radially symmetrical or not symmetrical at all. Cnidarians are radially symmetrical, but these lophophorates are bilaterally symmetrical. They also had a coelom. We talked about this as a body cavity. So coelom is a fluid-filled cavity that acts as a container for internal organs. It also provides a hydrostatic skeleton and rigidity against which muscles can push. And this allows greater mobility. So this is what allows brachiopods to burrow into the sediment. this ability to push using their coelom, this hydrostatic skeleton. So lophophorates also have specialized tissues and true organ systems. So they have tissues like the cnidarians, but they also have true organ systems, which is an evolutionary advance from the cnidarians, or a next adaptation. They also have a one-way digestive system, so they have a mouth and an anus. So their mouth only takes in nutrients, and then their anus is where the
does what it's supposed to do.
yeah, it excretes the waste products. So we talked in lecture about the evolution of the lophophore. So current molecular evidence suggests that brachiopods and bryozoans do not form a monophyletic clade. So they exist within the superphylum Spiralia, which also includes the mollusks. So it appears that the lophophore did not evolve parsimoniously. So either the lophophore evolved independently in brachiopods and bryozoa, which seems a bit unlikely because it's a very complex organ, or the ancestor of bryozoan, brachiopods, and mollusks had a lophophore, and then mollusks lost the lophophore.
Annelids are in Spiralia as well.
Yes.
So they lost it too.
Right. So we didn't talk much about annelids, but because they don't leave such a great record. But what could have happened is that the ancestor of the last common ancestor of spiralids had a lophophore. It was retained in bryozoans and brachiopods, and then lost in the mollusks and annelids. So there are two competing hypotheses for the evolution of the lophophore. We used to group everything that had a lophophore into a monophyletic clade, but that doesn't seem like it's the case.
Because it's such a specialized structure, it makes you wonder what kind of selective pressures occurred to lose it.
Yeah.
And then, not only to lose it, but for that loss of what was such a great adaptation for two groups of organisms, the loss of that leads to insane diversity in mollusks. It's very strange. It's something to think about.
It is strange because mollusks are an incredibly successful animal phyla. They have the second most number of species after only arthropods, and so if that diversity was only you could only attain that diversity by the loss of that lophophore, which seems like a really effective filter feeding organ. That's a puzzle.
So they freed them up to do something else.
Yeah. All right. So let's talk about the two animal phyla that we talked about. We talked about brachiopods. So brachiopods are also known as lamp shells. They have this two mineralized skeletons, so they're bivalves, but one valve is larger than the other. So one valve is larger than the other, and then the plane of symmetry runs right down the middle between both the valves. So that's how we can tell the brachiopods from the bivalves. So they used to be lumped with the bivalves, but the bivalves are a separate lineage because, for the most part, they have two shells that are mirror images of each other.
So the brachiopods, if they open like that, have a plane of symmetry here. The bivalves and the molluscans, if they open like this, have a plane of symmetry here.
Yeah. So that's how we can tell a brachiopod from a bryozoa. I'm sorry, that's not true. That's how we can tell a brachiopod from a bivalve. So brachiopods, the plane of symmetry runs through the valves, and then bivalves, they have two mirror image shells. So, but one of the brachiopod's valves is larger than the other, and that's where the pedicule extends from that valve. So usually the ventral valve is larger than the other, and the pedicule attaches to the ventral valve and then grows out of the valve to anchor it in the sediment. So brachiopods are marine filter feeders, and they are sessile. They don't move. But they have adopted a variety of different lifestyles. So some burrow into soft sediment, others sit at the sediment-water interface. They were the most abundant and diverse invertebrate group in the Paleozoic, but they suffered a great loss in diversity at the end Permian mass extinction. So there are still some brachiopod species alive today, but they live in cryptic habitats. So they're very small. Many brachiopod species are very small in tropical environments. And in polar regions or temperate regions, they may be larger, but live in these, they're hard to find.
But we have both lingulate and non-lingulate brachiopods still alive. So we have those with a large pedicle muscular foot that they use for burrowing, the lingulates. They're still around.
Yeah.
And then you have the more traditional lamp shell type that will attach to a substrate.
Yeah. So we talked about lingulates in lecture. So brachiopods are divided. We talked about both of them in lecture. But brachiopods have traditionally been divided into two classes, inarticulate and articulate. So now we know that those aren't true. Those groups don't reflect our evolutionary history, but traditionally they've been divided in those two, inarticulate and articulate. So inarticulate brachiopods do not have a mineralized hinge holding the two shells together. And this is where the lingulates fit in. So lingulates are, the lingula is a genera of brachiopods that has remained morphologically unchanged since the Cambrian. So it may be the oldest living genera of animals.
Oh, I've heard that, yeah.
Yeah. So inarticulate brachiopods are more common in the Cambrian, and they're much less common today. Articulate brachiopods make up 95% of all genera, so they're much more diverse through time. They have calcite shells, whereas inarticulate have shells made of apatite, and so calcium phosphate and chitin. And so brachiopods can live they can burrow into the sediment like lingula, or they can live at the sediment-water interface. So they have different modes of life. So let's talk about bryozoa. So we talked a little bit about bryozoa, and they are also lophophores. They are known as moss animals, colloquially. This is the only animal phyla that is exclusively colonial. So individual animals called zooids live in larger colonies called zoantharia, and they are marine filter feeders using their lophophore to extract nutrients from the water column. They are also the only animal phyla that did not evolve in the Cambrian. We talked about that earlier. So they have a one-way U-shaped gut, and let's see, I said that. They are found at all latitudes, from tropical environments to polar environments, and they may live down to 5,500 meters water depth. They have either attached, so forming these large colonies that are attached to the sessile benthos, or they're planktonic as well. Or they may encrust, so they may have these encrusting. We saw some of those fossils of these bryozoans that are encrusting brachiopods.
You'll see a lot of this on the field trip.
Yeah. So the morphology of these colonies, or these zoantharia, is not genetically controlled. So if you grow bryozoa in different environmental conditions, you're going to get different morphologies.
I did not know that.
Yeah, so temperature seems to be particularly important. So depending on the temperature, you'll get branching bryozoans, or massive bryozoans, or chain-like.
But the same species can form different structures. Yeah,
so the same species forms different structures in different environmental conditions.
What's that called, Andy?
That's called ecophenotypy. So
it's an ecophenotypic response.
It is. That's why taxonomy of these things, you really have to use thin section to get detailed taxonomy of
because if you did it on gross morphology, you would, and these things didn't have soft parts, so you couldn't do molecular analysis. You may end up taking different species that form the same colonial shape and lumping them into the same taxa.
Right.
And really, it's one species. You've got lots of different species converging on different shapes depending on the environment.
Right.
It's important to consider when you think about the fossil record. Is how can we be sure about species delineation?
Yeah. So to really get down to species identification in bryozoa, you have to do thin sections and then look for the morphology of these individual zooids and the openings in the zoantharia where the individual animal lives. Okay, that's all I've got to say about the lophophorids.
Right, let's do mollusks.
Let's go on to mollusks. So mollusks are within the Spiralia, but they do not have a lophophore. They are the most common group in marine environments, and they're the second most diverse animal phyla behind arthropods. They include clams, snails, chitons, scallop, octopus, squid, abalone, oysters, bivalves, and ammonites, and nautiloids. So a wide diversity of mollusks. They also have wide environmental tolerances. They can live in any aquatic environment, and gastropods, which is a class of mollusks, have invaded the terrestrial realm. So they're bilaterally symmetrical. They have a coelom. They are unsegmented. They're covered by thick, fleshy mantle, which secretes an aragonitic shell in almost all mollusks. So the shell in squid, this aragonitic shell is internal, so covered by soft tissue, and in octopus, they've completely lost this shell. Although in the embryonic development of octopus, you do see this very thin aragonitic shell forming and then dissolving.
It's worth looking for pictures of baby octopi just because they're really cute.
Yeah. And, yeah. So the head has sensory organs. They have a feeding organism known as a radula. They have a foot used for locomotion. And they have a visceral mass, which is an enclosure for their internal organs. So this is related. This is the same, similar to the coelom. So it's the visceral mass is the part of the animal that's the coelom.
It's guts, really.
Yeah.
It's just basically where it's keeping most of its internal organs.
Yeah. So coelom is an internal opening that you can store organs, to store organs. So when you try to reconstruct the hypothetical arch mollusk or the common ancestor for all mollusks, you're going to look for these characters. You'll look for a head with sensory organs, a foot used for locomotion, a radula for feeding, a mantle to secrete this aragonitic shell for protection, and this visceral mass, which is the enclosure for the internal organs. So mollusks are difficult. It's difficult to come up with a list of characters that every single mollusk share, but if you go to the base of the mollusk phylogeny, so the last common ancestor of all mollusks, they probably shared those characters. So all class of mollusks are known from the Cambrian except the scapopods. Scapopods are those tusk shells, but we're not going to talk too much about those.
Yeah, confusing.
So we talked about 3 classes of mollusk: bivalves, gastropods, and cephalopods. So we're going to briefly talk about those 3 classes. So cephalopods they have a well-developed head with tentacles and eyes. So these include modern squid and octopus. They also include modern nautiloids, and they include the extinct ammonites. So this class is predatory. In fact, the nautiloids were the first major predators in the Paleozoic. They also are very mobile, and so they use jet propulsion for locomotion. So you have to be mobile and have these well-developed eyes, so for binocular vision if you're going to go chasing mobile prey. And that's why they also have this well-developed brain. So squid in the invertebrates have the most well-developed brain of any invertebrate animal.
And the largest eyes. Architeuthis, the current largest invertebrate, the giant colossal squid, has the eyes the size of dinner plates. Yeah, big.
And these are all adaptations that you need if you're going to pursue a predatory lifestyle. So ammonites were cephalopods that experience a great radiation and diversity in the Mesozoic, and they're often used as biostratigraphic indicators in the Mesozoic. So that's cephalopods. So we also talked about bivalves and gastropods. So following the end Permian mass extinction, bivalves and gastropods replaced brachiopods as the dominant marine invertebrate of the benthic environment, so at the sediment-water interface. And bivalves and gastropods have also experienced a real increase in diversity all throughout their evolutionary history, and especially since the end Permian mass extinction. So gastropods are snails, so they are in all aquatic environments, and they've also invaded terrestrial environments. They exhibit this coil. They have an aragonetic shell with no septa. So cephalopods, many cephalopods, have septa that separate the chambers of their shells, but gastropods have just one shell, so they're univalve, and it's not separated by septa. Their body has undergone torsion, so their head is on the ground.
The butt's above their head.
Yeah. So it's like they're opposite of us. They use their radula. It's been modified to scrape. So many of them are grazers.
Detritivores, right?
Yes. So gastropods are all different kinds of feeding. So they're detritivores, using their radula to scrape. They're grazers, using their radula to scrape microorganisms. And also some radulas have been modified into this drill that can drill through bivalve shells, for instance, and they're predators. They use their shell for drilling. We saw Conus, which is the killer snail. It uses its radula to drill through to eat bivalves and other invertebrates. So that's all I have really to say about gastropods. So we also talked about bivalves, which is another class of mollusks. So they have a hinged aragonetic shell that is bilaterally symmetrical. So their two shells are mirror images of each other, except in oysters, which is a bivalve where the shell is made of calcite and they're not mirror images. They have lost their head, so only the presence of their head is indicated by their sensory organisms. And their foot has been modified for burrowing and all kinds of other environments. So we can actually tell the mode of life of bivalves by their morphology. So this is really useful in paleontology because we can tell whether it was a burrowing bivalve or if it lived at the sediment-water interface based on their shell morphology. So they have highly derived gills, so large gills, and they have abductor muscles that allows them to open and close their shells.
That's what you eat in a scallop.
Yeah.
Nom, 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 nom.
They have delicious abductor muscles. So scallop pecten are also the only nectonic swimming bivalve. And some, like the giant clam, have endosymbiotic algae. So that's all we got on mollusks. Okay, so let's move on to arthropods. So arthropods are the most abundant and diverse animal phyla, and they have been the most abundant and diverse animal phyla ever since their evolution in the Cambrian.
They basically win at evolution, right?
They win at evolution. Yeah, so we want to think about, okay, why are arthropods so evolutionarily successful? And we discussed some of that. So arthropods have an external multi-layered exoskeleton This provides protection, but it also serves as an anchor point for mussels. They also have a high reproductive rate and rapid evolution, allowing them to fill many niches very quickly. So we also talked a little bit about their body plan. They have this segmented body, so they have a lot of evolutionary flexibility to modify their body, either to fuse segments or to divide into further segments, allowing more adaptations to different environments.
So the division into segments is metamerism, and the fusion is tagmosis.
Right. So arthropods have a segmented body with jointed legs. So arthro is jointed and pod is legs. So arthropods, jointed legs. They have a chitinous exoskeleton, which is reinforced with calcite in many groups, giving them a really good fossil record.
Mostly marine arthropods because of the free salts that are available in seawater. Terrestrial arthropods don't have that kind of free salt available in a terrestrial environment, and so their exoskeleton is pretty much entirely chitin.
Right. So chitin is less likely to be preserved in the fossil record than calcite, but we know a lot about the evolutionary history of arthropods because we get really great soft part preservation in these lagerstätten. So we know a lot about arthropod evolution. They grow by molting. So this chitinous exoskeleton provides great protection, and it's flexible, but to grow, for the animal to grow, it has to shed its old exoskeleton and then regenerate a new one.
It's not being locked inside a spacesuit as you grow.
Yeah.
You have to get a new one every time it gets a bit too tight. You have to get out and get a new one.
Yeah. So the basal body condition for arthropods was 3 segments: the head, thorax, and abdomen.
And when you have those 3 segments, it's called a tripartite body plan.
Right. So there are 5 subphyla of arthropods, and so if you can keep track of what's happening to those 3 segments of the body, you'll have a good framework for understanding the evolution and the diversity of arthropods. And they're incredibly successful. They're in almost any environment on Earth, you'll find arthropods. And some of the first animals to recolonize an area after a disaster are arthropods. All right, so those 5 subphyla are trilobite, hexapod, crustaceans, chelicerates, and myriapods. So we're going to talk briefly about each of those 5 subphyla. So trilobites are exclusively marine, extinct organisms. They went extinct at the end Permian mass extinction. But their body is divided into 3 parts lengthwise. So they have the axial lobe and then 2 pleural lobe, and that 2 pleural lobes, and that's what gives them their name trilobite. But their body is also divided into those 3 segments, like many arthropods. We call the head region the cephalon, the thorax is the middle region, and the pygidium is the tail region. Good, I pronounced that right.
I'll give you a pro tip. The cephalon, so Ceph is what? Greek for head.
Okay.
So every time you see C-E-P-H, it relates to the head of an organism.
Cool. So we got cephalon, thorax, pygidium. Pygidium, thank you.
Oh, you're on a roll as well.
I know. So they have a calcite exoskeleton that gives them a really good fossil record, but they have no specialized mouth parts. So most trilobites were benthic organisms. They were either scavengers or grazers, and some of them could swim short distances, so they were nectobenthic organisms. So most of them were just on the seafloor bottom, and there were a few that were planktonic, and some were actually nectic. But most trilobites were benthic organisms.
They just scuttle about on the seafloor looking for detritus to shove in their hyperstone.
Yeah, to eat. So yeah. So what caused the extinction of trilobites? Well, there was a global cooling and a sea level drop, which probably caused their extinction, and also the appearance of predators like cephalopods to eat them contributed to their decline. So the end Permian mass extinction was like the death knell in the coffin of trilobites. They were on an evolutionary decline up to then. So they had been their heyday, so the highest diversity of trilobites were in the Ordovician and the Silurian, and since the Silurian they had been declining in diversity. And then they finally went completely extinct at the end Permian mass extinction.
It's probably not a coincidence that that is exactly about the time that large crustaceans appeared to dominate fauna.
So, crustaceans are the dominant marine arthropod today. They include lobsters, crabs, shrimp, ostracods, clam shrimp, barnacles. So they are biramous. They have, just like trilobites, they're both crustaceans and trilobites are biramous. So arthropods have two appendages on each segment, so one on each side. And then biramous means you have Y-shaped appendages.
So put your arm out. I reckon we can
make a biramous. Woo!
Like that.
Woo! So if this is their appendage here.
They have a separate one coming out down here.
Yeah. Woo! So this might be a walking leg, and that might be a gill. So biramous, it splits into two there. Crustaceans and trilobites are biramous. So crustaceans have two antennae and then three appendages after their mouth, right?
Yes.
Posterior to their mouth. Mm-hmm. And they also have a segmented abdomen. So they are the dominant marine arthropods today. All right, so hexapods are the dominant terrestrial group of arthropods. This includes the springtails and the insects. So springtails have, they don't have wings and they have covered mouth parts. But they are not particularly speciose. They're not particularly diverse. Insects are, they are a class within hexapod, and they have exposed mouth parts and they have wings. They're an incredibly diverse group of arthropods, and they're the most diverse group of terrestrial animals living today. So hexapods have a head, thorax, and a tail. Insects have mouth parts exposed, wings, simple eyes, and their abdomen is differentiated. So we talked about some evolutionary trends with insects. We see this coevolution with flowering plants. So we see the rise in diversity of flowering plants at the same time as the rise in diversity of insects, because they have those specialized pollinator relationships where the insects pollinate the flowering plants. We also see this coevolutionary relationship between flies and spiders, where increase in diversity of spiders, it coincides with an increase in diversity of flies, but that's not because they have this mutualistic relationship. That's because they have this predator-prey relationship. And we also, in the Carboniferous, we see these giant insects because the atmospheric oxygen levels were really high. So the last two subphyla, we've got myriapods. These are centipedes and millipedes and their allies. They are uniramous. They are the first land animals. We have body fossils from the Silurian and trace fossils from the Ordovician or perhaps even the Cambrian on land. So myriapods form these very distinctive trails due to their, they have so many legs. So myriapods have a small head, a small tail, and then their thorax is just these repeated segments, each with a pair of assemblage of uniramous appendages on each segment.
They have very, very distinct. They leave very distinct traces. They're especially in millipedes. The millipedes have a sinusoidal leg movement, so each pair of legs moves up one after the other to create like a wave-like pattern, and that leaves very distinct traces in wet media.
Yeah.
It's easy to see in the fossil record.
So we see those terrestrial trace fossils perhaps as early as the Cambrian. So
they're aqueous, though, right, in the Cambrian?
well, they're they're terrestrial, so they're
Right.
they're in moist environments. But so the Cambrian trace fossils are a little bit speculative, a little bit more equivocal, but so definitely trace fossils from the Ordovician and maybe as early as the Cambrian. But body fossils from the Silurian. So first animals to invade land, myriapods. And the last subphyla of arthropods are the chelicerate. So these are uniramous, and they have chelicera, which are these pincers. A lot of their first appendages, a lot of time in their mouth, and then they have another pair of appendages, and then 4 pairs of legs. So they have
they have the chelicera, is the first pair. The second pair is called the pedipalp.
Pedipalp, thank you.
The second, and that is the pincers in scorpions are the pedipalp, and in spiders, if you ever catch a really nasty, massive, hairy, mustachioed, heavy-breathing, hat-wearing, sunglasses, face spider in your bath, then you often see right at the front of the mouth it has like 2 short club-like feelers. So they're pedipalps. So, like what I said earlier on, it's the pedipalps have been utilised for sensory appendages in spiders and for manipulative appendages in scorpions. Again, even within a subgroup, the Chelicerata, it's a testament to the flexibility of the body plan. It's amazing.
Yeah. So, chelicerates are arachnids, so including spiders, scorpions, ticks, mites, horseshoe crabs, and eurypterids. So eurypterids are these extinct predatory sea scorpions.
No actual real relation to scorpions. They just look like them.
Yeah, so they're not
reside in the same group.
Yeah. The chelicerate combine their head and the thorax to form the prosoma. So they have 2 body segments, the prosoma and then the opistosoma. And then actually in ticks, those two fuse to form just one body segment. So,
Tagmosis.
Yep.
To fuse.
And then all right, so that's all I got to say about chelicerates. Yeah.
Apart from chelicerates, some of them have book gills, so they have two methods of respiration. What else?
How do the book gills allow them to respire?
That is again diffusion across a moist surface membrane. So it's like having, if you can imagine having gills like a fish that you can keep somewhere moist. So they have like a box on the back part of their abdomen with the gills in, with a flap, and in there they keep that moist, and it allows them to pass small amounts of air over it and diffuse oxygen into the blood that way, as well as having tracheal systems as well.
All right. Well, that's arthropods are the last topic for this exam. So, I hope that this video is helpful to you. We ran a little long, but I hope it is helpful. So, please bring in your questions on Wednesday.
All right. Good luck.
See you later.
See ya.